Yes, good. We're recording. Good morning, everyone. How are you? Semester is almost, oh boy, geez, just jumping right into it there. Very tired. Yes, I think we're all tired of everything. Um, but the semester is almost over. How do I make this thing go away? Go away. Let me see if I hope this doesn't stop recording it if I make this thingy go away. Let's just see. Nope, still recording. Okay, cool. Yes, semester is almost over. Our last class is a week from today, believe it or not. It's crazy. But it's good. Just double check that day before I say that to make sure. Pretty sure that. Come on, phone. Yeah, wow. Yes, so we have today's lecture and then we have two other lectures. Yeah, so thanks guys for doing the Angiosperm documentary assignment. Hopefully you guys enjoyed doing that. I like that documentary. I think it does a good job of covering some interesting information in a way that's kind of digestible. I like how it makes it it's like very dramatic music and things like that. Yeah, people are asking, okay, so dissertation defense is going okay. I defend on Monday, so I'm just working on the presentation right now. And I have a practice of that on Friday, so looking forward for that to be done so I can get some real sleep and be more like a normal human. Phenology assignment, yeah, still haven't really thought about it too much. A few of you have sent me your phenology assignments already, which I appreciate. Let me think about it. I know technically it was due the last day of class, but let's push that deadline for now. Probably what it will end up being is something like that you guys can send me what you were able to do in those first few weeks. But let's hold off on that now so we can get through the material, and I will make a definite decision sooner than later. But the deadline will get pushed, so don't worry about it too much right now. All right, let me pull this up so I have where I'm going. Let me move this out of the way so I can see. Okay, cool. And let's just get into it. How do well, I'll just move this here. Okay. Yes, we're finishing up this semester with the angiosperms. We started talking about them the last time that we met. We talked just a little bit of an intro about the angiosperms. Today we're going to continue with those, more about growth, which obviously is my most favorite topic, and then we'll finish off today talking about these different evolutionary lines in angiosperms. So just an FYI that the depending on what source you're looking at, so if you're looking at the lecture slides, I'll be calling the angiosperms Magnoliophyta, but you may see terms in the lab saying Anthophyta. It's the same thing. However, Anthophyta is kind of considered outdated now, and we'll talk more about why that is toward the end of the lecture. Oops, I can't see this because this is in the way. I'll move this away here. Yes. So we've already talked about this a little bit in terms of growth in angiosperms. So we talked a bit last time and probably the times before that about these primary meristems that give rise to the primary growth. So if you were to, we looked at this last time, look at where that apical meristem is in the shoot, you would see the primary meristems. If you were to do a cross-section through the tip and look at it under the microscope slide, you would see these primary meristems, which are the result of this apical meristem, right? So here, just as a little recall, here's the apical meristem right in the middle at the very, very tippy top of the shoot. And that apical meristem is going to be giving rise to these three primary meristems, which in turn give rise to primary growth tissues. And we've already talked about these lateral meristems, which are the what happens during secondary growth, remember, because to get wider as versus longer. And these two lateral meristems are called the vascular cambium and the cork cambium. Vascular cambium gives rise to your secondary xylem and secondary phloem. Cork cambium giving rise to the cork, aka phloem, and the phloderm. And this is, I know you've seen this before, but I'm just putting it all together to show you, here's those primary meristems again, which we just saw 2 slides ago. Here's those secondary meristems again, the vascular cambium and the cork cambium. And this main reason I'm bringing this up again is just to keep it in mind that depending on where you're looking at in the plant, you will see either primary growth tissues or secondary growth tissues, or maybe a little bit of a mix of the two if you're transitioning from primary growth tissue to secondary growth tissue. So this slide is just showing you if you were to do a cross-section at the tip where the shoot system is trying to lengthen, of course you're going to see primary growth tissues there, right? Epidermis, cortex, primary xylem, primary phloem, and in the case of this pith, right? But if you were going to do a cross-section toward the bottom of the plant where that secondary growth tissue is already starting to happen, you'll see these secondary growth tissues, right? You'll see the secondaries, but that's the point of this slide. This is not, this plant on the left here is not super old, right? It's not super wide. It's not like a ginkgo that is, 100 years old or whatever, that's 4 feet wide. It's just a little plant that's starting to make that secondary growth tissue. And so even though you'll see secondary phloem, vascular cambium, right? Secondary xylem, paradermic cork cambium, you'll see when it's starting to transition, you will still see some of those some of those primary growth tissues, right? Like primary xylem, primary phloem, pith, cortex, etc. And then eventually, this will be completely composed of secondary growth tissues. Just a little bit more recall. The primary meristem that produces primary xylem is the procambium. The lateral or secondary meristem that produces secondary xylem is the vascular cambium. So I'm just putting those together because both of the meristems that produce xylem have cambium in the term, right? Procambium is the primary one, leads to primary xylem. And then vascular cambium is the lateral meristem that leads to secondary xylem. So today we're going to talk a little bit about xylem. That's why we led into these slides. We're setting it up to talk a little bit about xylem cells. So in vascular plants, the main or principal cell type of the xylem is called tracheary elements. And there are 2 kinds. There are tracheids and, sorry, I'm just reading the chats that I didn't see before. There are tracheids and there are vessel elements. So if you're talking about either of them, they have certain things in common. So remember, these are cells of xylem. Cells of xylem is what we're talking about right now, right? Tracheary elements. 2 kinds, tracheids and vessel elements. If you're talking about either kind, they have certain things in common. They're both elongated, so long cells. They're both dead at maturity. And they may or may not have pits, depending on where they are in the plant. So some plants, so we're talking about xylem cells here. So some plants only have tracheids. As I often say in terms of evolutionary time, the more primitive or older lineage plants have tracheids. Tracheids are the ancestral cell type of xylem. So what does that mean? If you were going to look at a cross-section of a stem of xylem that has from ferns, some of the tree ferns, or the lycopods, or the horsetails, right? Those earlier vascular plants, the spore-releasing or spore-shedding vascular plants, they would have tracheids. If you would do a cross-section through a gymnosperm, they would have tracheids, because tracheids are the older cell type, or the more ancestral cell type. You might remember when we looked at conifers, when we started to talk about secondary growth tissues more in depth, we talked about how you can view the wood different ways depending on how you cut it. So this is a piece of a trunk here showing you secondary growth structures. And depending on how you cut it, you have different views of the cells, right? So like if you cut it, you're going to see the cells in different ways depending on how you cut it. So if you were to cut the tree down so that it's just a stump, and you were to look down at that stump, this is what you would see. You would see a cross section or a transverse section. And that means that you would see all the cells from the top. So if they're like a straw, you would see them as if you were like looking down the straw, right? Because all the cells are going, remember, these are tracheid cells and their job is to move water. So where are they moving water? From the roots to the shoots. And so they are oriented up and down, right? Vertically, because they have to move, they grow up, right? They don't grow back. So they grow up. And so they're kind of like the shape of a straw. So if you could cut the tree down and look at the top of the stump, all of the cells would appear like these little, all these little holes here. Those are kind of like the tops of the straw. And that would be if you had a cross section. However, if you were to cut the tree in half instead of like down, right? Like if you were to saw the tree in half, this is what you would see. This is one of the views you could see, right? Of a tangential section, right? In this case, you're not seeing the tops of the straws, right? You're kind of seeing the sides of the straws, but then now you are also seeing those cells that run horizontally in the stem that we talked a little bit about last time, the wood rays. And again, if you were to cut it this way, now instead of seeing the end of the wood rays, right? So let me do this. Right, I'll change colors because that's already red. Let's pick yellow. Okay, so here, right, this is a wood ray. This section here is a wood ray. If we were to cut the tree in that way, we could see one of those wood rays going horizontally from the inside of the trunk to the outside of the trunk. But if you were to do a tangential section, right, this is what the wood ray would look like here. See, you would only see this part of it. Since we're drawing things, might as well just show you what I am talking about also. So like here, remember we were just talking about tracheids, which are a cell type of xylem, right? If you were looking at the cross section up here, here's the top of a tracheid, right? Like the top of the straw. But in this radial section, you're seeing kind of this side part of that tracheid, right? Which is then obscured by this wood ray. Here is that top of the tracheid over here, and here is the... So I'm just showing you this so that you can see, like, trees, plants with secondary wood, they're three-dimensional structures. So depending on how you cut them and how you're looking at them, you might see different things. Okay, enough annotating for now. Yes, all right, let's move on. So talking about cells in xylem, we're talking about tracheids, which are the older or more ancestral type of cell type in xylem. And this is what I was just trying to say and draw, is that tracheids can have two orientations in the secondary xylem. They can grow vertically or they can grow horizontally, as is the case in the wood rays. So now instead of a block of wood, we're looking at a microscope slide of wood, right? Remember, wood is another way of saying secondary xylem. So here let's, so this is a cross-section view. So just to remind you, this is a cross-section or transverse view. Now, instead of looking at it on a wood block, we're going to look at it under the microscope slide. So here you go. And again, you're seeing those tracheids, you're seeing the tops of them, right? Like a bunch of straws. So each of these things here, each of these little circles is the top of a tracheid. And then you can start to see some other characteristics of the wood, right? Like early wood, which is that spring wood you guys probably talked about in lab. So spring wood or early wood, the tracheids are much bigger. Late wood or summer wood, when water is not as available, you'll have the tracheids being much more small and condensed. And both of those make up an annual ring. Just to stick with our color scheme from the previous slide, right? So here's wood rays. Here's a ray. Come on, keep going. There's a wood ray, right? Remember, the wood rays radiate like a radius out from the inside of the stem to the outside of the stem, right? So there's your wood rays using our cross-sectional or transverse view. And each of these little circles each of these is the top of a xylem, top of a tracheid, which are the cell type that makes up the xylem. So anyways, yes, just to use the same colors we saw in the previous slide. Now we're looking at a radial view under the microscope slide, and similar. So this is tracheids having both your horizontal and your, yes, are wood rays composed of tracheids? Yes, they are. Yep, anything in the wood. And again, right now we're talking about the wood of gymnosperms, the wood of lycopods, although nowadays they don't really get super woody, but even like the, yeah, we're talking, think about it mostly in the gymnosperms right now. That would be the easiest way to think about this. But these are plants that only have tracheids as versus the other cell type, which we're going to talk about in a second. But yeah, anything in the wood is tracheids. So just to use our same color scheme as we did before, because now we're looking at a radial view. One thing that's nice about the radial view is that you can easily, oh, nope, that's bad. Nope, oh geez. Annotate is like the, okay. Radial view, so you can see rays, wood rays. Here's wood ray here, right? Here's part of a wood ray here. Here's part of a wood ray here. Each of those are made up of tracheids. And then those are horizontal tracheid cells. And then each of these in this radial section is a vertical tracheid, right? Elongated, dead at maturity, sometimes having pits, right? Get out of annotate. There we go. Okay. Make sure I didn't miss question. Okay. And then lastly, this is that tangential view. Rather than explain it again, I'll just use the colors to show you. Last slide, we saw the wood rays going across because that was the radial view. Now we're seeing the edges of the wood rays in our tangential. So each of these is the end of a wood ray or a wood ray cut through. And each of these long things next to it, right there, all these things here, are those vertical tracheids. Okay. I am going somewhere with this. All right, so now we can move on from looking at cells and talk about it a little bit. So talking about tracheids, tracheids are cells that make up xylem. Tracheids have pits, okay? Tracheids have pits. The pits are what allow water to pass from cell to cell. So remember, that's the job of tracheids, is to move water inside. Sorry. That's the job of the xylem, is to move water inside of the plant. If the xylem is made up of tracheids, the job of the tracheids is to move water. Tracheids have pits, allowing the water to go from one tracheid to another tracheid. The water that passes from a tracheid to another tracheid has to go through a membrane that's in the pits. It's kind of like this part of the cell wall. So the water can't just go from one tracheid to another tracheid. It has to actually pass through cell membrane. And the other thing about tracheids is that water can't go vertically from one tracheid to another tracheid. The water can only pass horizontally from one tracheid to another tracheid via the pits. So let's look at this. So let's look at the radial view first, right? So these are the vertical tracheids that are running up and down in the trunk. And we could see the pits. The pits are these circles. And we can see the membrane, which is in the center of that pit. And that's the thing that the water must go through if it's going to go from one tracheid to another tracheid. Here is our cross-section view, or transverse view, from the top if you were to cut the tree down. And you could see, so each of these circles is a tracheid cell, and you can see that the pits are between certain tracheid. So this tracheid cell has a pit right here between this tracheid cell, and water from this tracheid cell could go through that pit to the other tracheid cell, but not vertically, so not upwards. Can only pass water horizontally from one cell to another cell. So that's tracheids, and that's primarily in the gymnosperms. But in the angiosperms, which are a little bit more evolved, they don't just have simple tracheids anymore. They have what are called vessel element cells and fiber cells. And so these are what tracheids evolved into. Vessel element cells and... Okay, so I have a question here. If they are vertical, wouldn't water travel faster and be more beneficial? Meaning, wouldn't it make more sense for water to move vertically from tracheid to tracheid instead of horizontally from tracheid to tracheid? Great question. You would think so. And yes, there are some advantages to being able to move. If you're wanting to get water from the root to the shoot, there are some advantages from being able to move water vertically. But as we'll see in a couple slides coming up, there's reasons why that could also be disadvantageous, and why this evolved, right? So good thinking, and that should become clear in the next few slides. So we talked about the tracheids before. Now we're talking about, sorry, something's parking outside. I don't know if you hear the beeping. And we talked about the tracheids before. Now we're talking about what the tracheids evolved into, which are in green vessel element cells and in purple fiber cells. First, we'll talk about the vessel element cells. So just like tracheids, vessel element cells are also non-living. However, unlike tracheids, the vessel element cells, as Gabriella was kind of thinking about, is like now the vessel element cells have a way to move water upwards, not sideways from cell to cell like the tracheids. Now the water actually does flow upwards. And the way that it's able to flow upwards is because that at the top of each vessel element cell, there's this plate, this perforation plate, which allows the water to flow through. Now, the vessel element cells still have some pits on the side, so they still are, in some cases, able to move water sideways, but the primary way the water moves through vessel element cells is now upwards instead of sideways through these plates. So here is what one of those plates might look like. This is a very simple perforation plate in that it has one opening. So this is literally just like a big hole at the top of these vessel element cells, big, giant, gaping hole. that water can move from one to the other. Yeah, I know, it's cool. But note that this vessel element cell still does have pits. It still does have pits on the side. But, I mean, come on. You got this giant opening at the top. And so the water is primarily moving upwards as versus sideways. This is also a perforation plate at the top of vessel element cell. However, this one is more complex. Kind of looks like a ladder. But it still serves the same purpose. It's this opening at the top of vessel element cell that allows the water to flow upwards from one vessel element cell to the other one. And again, notice it still contains pits on the side, but not the primary way to move water. Instead, you have these big giant plates. So as Gabriella was kind of thinking about, yeah, there's like, this is great because now we don't have to move water side to side. Now we can just move water up. So like, awesome, no problems. But there's pros and cons to having these vessel element cells. The pro is, yeah, sure, you're more efficient, right? You don't have to move water just sideways from cell to cell now. Now you actually can move water straight up. So that is a pro, more efficient. However, it's a little bit more risky to have these vessel element cells. Part of the reason why it's more risky is because with the tracheids, when you had to move water sideways from trachea to tracheid, the water had to actually move through a membrane. So if your water had other things in it that wasn't good to move from one cell to another cell, that membrane can kind of prevent that from moving from one cell to another cell. So if there was some sort of pest or thing that was bad for that cell, it would stay localized in the trachea and not be moved to another cell because it would have to go through a membrane. But now in vessel element cells, there is no membrane. So that could be a little risky because it's just an opening, you know, and things can just flow right through from cell to cell. But the riskier thing, if that's not risky enough, is that you can have air bubbles form, which could effectively turn a vessel element cell to be like not usable anymore. So if you if you have, this is my like little wand thing for my niece. Ooh, but it can serve the purpose for right now. So if you're, like, say this is a vessel element, say this is a tracheid, right? Or say this is a vessel element cell, right? And the water can move from one to the other, right? If an air bubble forms, like let's say an air bubble forms right here, that's it. You can't overcome that air bubble. And so this vessel element cell would just be non-functional anymore because the air bubble would prevent it from moving up. But if this were a tracheid and you formed an air bubble right up here, you could still move water to the other tracheid below that air bubble. So that is another reason why it's a little more risky to be using vessel element cells, because if specifically, and this is why I think conifers have stayed with having tracheids, if you undergo freezing, so if you're in freezing temperatures, the mail is arriving. If you're a plant, like if you're growing in the circumboreal forest where you experience freezing temperatures quite often, freezing temperatures, I don't know if you've ever done something like this, but if you have like a water bottle that's completely full and you put it in the freezer, water expands when it gets frozen. And so it would get larger. Then when it would start to defrost, right, the water would go back down. In xylem cells, you have water. And so if the water freezes, you run the risk of creating these air bubbles when the water, yeah, the mail. When the, can you guys hear it? It's so loud. But that's the whole point. The point is that it's risky. Even though these are more efficient cells, it's risky because water doesn't have to go through a membrane anymore. And the tracheids, air bubbles can form, which would just make them not efficient anymore. And then we have to talk about these fiber cell things. This is still talking about parts of the xylem. We just talked about tracheid cells. Then we talked about vessel element cells. Now we're talking about fiber cells. Fiber cells, just like tracheids and just like vessel elements, are not living. They're dead when they're functional. But fiber cells' main characteristic is that they have these like really thick walls, right? Really thick cellulose walls. And the presence of fiber cells in angiosperms is why we call the angiosperms hardwoods and why we call the conifers softwoods. So this is, I think, a question in your lab. Like, what's the difference between a hardwood and a softwood? Angiosperms are hardwoods because they have fiber cells. Conifers are softwoods because they don't have fiber cells. Now, I think I've said this before, maybe not, but it doesn't mean that conifer wood isn't hard, right? If I had a piece of conifer wood and I hit you on the head with it, it's still hard, right? It's not like hitting you with a leaf, right? It's still hard wood. But when we use the words hardwood versus softwood, we're talking specifically about presence or absence of fiber cells. All right, I like this slide because it kind of puts it, this slide and the following slide kind of like put this all together because it's like, why is Rebecca talking about cells so much? Okay, so this is a comparison of the different cell types that we've just talked about. On the left, you see the wide vessel element cells. So A and B are wide vessel element cells. Notice they still have pits, right? The spots are pits. They still do have pits on the side, but the main way that they move water is through these giant perforation plates at the top, right? Next to those wide vessel element cells is an example of a narrow vessel element cell, so they're not always wide. And then next to that is a tracheid cell, which is D. And then examples of fiber cells are in E and F. But just look, like, so if you're trying to move water, look at A and B. A and B seem like better ways of moving water overall than D, right? D is kind of puny, doesn't have big openings at the top. So that's why I think the majority of, that's why once we get to angiosperms, they have these vessel element cells instead of just tracheids. This next slide is cool too because it just shows it in a different way. So the order is reversed, right? Here from left to right, we have vessel element cells and then tracheids. Here from left to right, we have tracheids and then vessel element cells. So starting on the right, here's our big wide vessel element cell, big giant perforation plate. This is more like the perforation plate we saw in the simple one. Oh, question, do fiber cells participate in water transport? No, they don't. They're part of the secondary xylem, and they serve for structural purposes, but they're dead on maturity, but they don't move. Part of the reason they don't is because if we can go back here, maybe there are some types of fiber cells that might participate a little bit, but in general, the fiber cells look like F. So in F, you can see doesn't have pits, doesn't have perforation plates, and so their primary purpose is structural and not conducting. So yeah, on the right here is those vessel element cells. You got an example on the first two here with just a simple perforation plate, a big opening, no obstruction. Next to that, a different type of vessel element cell with the more complex type of opening, like that ladder type opening we saw on that microscope slide a few slides back. And then, of course, tracheid, right, which just has your pits. Okay, hang in there. So we talked about the tracheids and the vessel element cells and the fiber cells. Those are all parts of the xylem. Now we're going to talk about the phloem briefly. Just like there's cells of xylem tissue, there are cells of phloem tissue. And the main or principal conducting cells in phloem are called sieve elements. And there are two kinds of sieve elements. There are sieve cells and sieve tube elements. So just to compare, so Sorry. Right. So just to compare, in xylem, the principal conducting cells are tracheary elements, and the two kinds are tracheids and vessel elements. In the phloem, the primary cells are sieve elements, and there's two kinds, sieve cells and sieve tube elements. So just like before, when we had the xylem, right, there are two kinds of xylem, tracheids and vessel elements. Tracheids are for the gymnosperms and the more primitive vascular plants. Similarly, sieve cells are for those primitive vascular plants and gymnosperms. Sieve tube elements are for the angiosperms. So when you're talking about these structures, the ones that have elements like sieve tube elements or vessel elements are the ones that are in the angiosperms. That's how I remember it. Okay, so here we have, this is a phloem. So this is, we're going to talk about first the sieve cells and then the sieve tube elements. So here are examples of sieve cells, which are in those gymnosperms and the more primitive vascular plants. Sieve cells are living, all of the tracheid cells and all of the xylem tissue we talked about before, fiber, tracheid, vessel elements, those are all dead on maturity. These are all living on maturity. So sieve cells are living, and they only have these little small pores, which are shown here in blue. And like the tracheids, sieve cells can only move sugars sideways from cell to cell, not downward. So remember, tracheids are moving water from the roots to the shoots. Xylem is moving water from the roots to the shoots. Phloem is moving sugars from the shoots to the roots. So sieve cells are the more primitive type of cell. And like the primitive type of cell in the xylem, which is the tracheids, the sieve cells can only move things sideways, right? Not straight down. So they move things sideways. I'm trying to draw comparisons from these so it kind of helps you guys remember it. Okay. Come on, wake up. Okay, so that was the sieve cells. These are the sieve tube element cells. And remember, so if it has an element in it, it means it's in the angiosperms. Similar to vessel elements, these are, they have these larger pores located at these plates that do now allow their materials to move directly vertically from cell to cell, in this case downward, because they're moving the sugars from the shoot system to the root system. So I wonder if I have a good, okay, yeah, we're almost done with cells. We're almost done with cells, I know. If I could sum all this up quickly, it's that you have xylem and you have phloem. You have two cell types in xylem. You have two cell types in phloem. In xylem, the cell types are tracheids and vessel elements. In phloem, the cell types are sieve cells and sieve tube elements. All the xylem ones are dead on maturity. All the phloem ones are living on maturity. The simpler xylem one is tracheid. Hold on one second. I just want to see. Oh. This is kind of weird, but hold on. I have to let in this. I have a package. 1 second. Sorry. Okay. Yeah. So, to what I was saying, in the simple plants, or sorry, not simple, in the older evolutionary plants, sorry, I just ran. The materials move sideways instead of up and down. In the xylem stuff, it's the tracheids. In the phloem, it's sieve cells. In the more advanced angiosperms, materials can move up and down, not just sideways. Vessel element cells, sieve tube element cells. Okay. All right. And just like before, this shows you a comparison of those sieve cells versus sieve tube element cells. Sieve cell on the left looks a lot like a tracheid. Sieve tube elements on the right with those special plates at the top and the bottom that allow the materials to move vertically. Okay, we're done with cells. We are. Phew, thank God. Oh, no, we're not. Crap. All right. Last thing is that, yeah, we're almost done with that. So remember, the cells in xylem are dead on maturity, right? The cells in phloem are living on maturity, but they also, even though they're alive, they lack nuclei, which is crazy. So it's like if you're a cell without nucleus, how are you technically alive? And I will show you. So with the sieve cells and the sieve tube element cells, they have these special friend cells that hang out next to them that are called companion cells. And they basically keep the sieve cells, sieve tube element cells alive. So here is, do I light it up? No. Here's just talking about these companion cells. So here you can see sieve tube elements, right here. And they're basically hollow. So they don't have a nucleus inside. Because if you're trying to move sugar through the plant, right, either if it's horizontal with the sieve cells or vertically with the sieve tube elements, you don't want a lot of cytoplasm and nucleus and all this other stuff inside of your cell because you have to move sugar. So they're basically empty, but they're kept alive basically by these companion cells which hang out next to them, and those cells have nucleus and are able to keep the cells alive. Okay, I'm so over talking about cells. There's a nice summary at the end of this lecture that kind of puts this all together instead of on lots of different slides, on like slides combining all this information together. But it's better to go through it so that you guys can understand it. All right, well, how's our time? We got a little bit of time. How many slides? All right, we'll see how much of this I can get through, but we're not talking about cells anymore. So, yay, flowers, phew. So, as a reminder, right, the angiosperms are the most diverse group of plants, and there's a ton of different variation in morphology, not only in the flower, but also in the form of the plants. So we learned in the conifer lecture that the tallest trees in the world are conifers, but angiosperms have some pretty tall trees as well. So these are the tallest angiosperm trees that are living today are eucalyptus trees that are located in Australia. And they can get to be about over 300 feet tall and over 70 feet wide. So that's super, super big. I mean, 70 feet wide is pretty crazy big. But you also have really, 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 really small plants. So these are plants that are in the family Lamiaceae. This is type of plant family that's located worldwide. We call members in this plant duckweeds, even though it's not the same as that azolla, which we also call duckweed. So remember, azolla is a fern, and these are angiosperms. It's another reason why common names aren't necessarily so great, because a lot of times people use the same common name to describe, yeah. So Masahiro says, oh, I have these as well. Yeah, they look really similar. They do look really similar, but you know how you know they're not the same? Because these are angiosperms, which means they what? What do angiosperms shed? Right? Or what does angiosperm? Yeah, they shed seeds, and so that means that they have flowers, right? So these, even though these look with the naked eye very similar to Azolla, which we also call duckweed, if you were to look closer, and we will see on the following slide, these actually bear flowers, as versus the duckweeds that we saw before of Azolla, they don't bear flowers, they're ferns. Yeah, I think their seeds don't last very long. They don't have a lot of resources to give towards the holding on to them. So I'm showing this thing on the bottom here to show you where Lamiaceae has been reported as occurring. Lamiaceae, similar to Azolla, is also used a lot in the aquatic, like in the aquarium industry. So unfortunately for that, similar to Azolla, it's been released into waterways via use in aquariums, and so it can become a bit invasive in places. And just to give you an idea of how small these plants are, on the left, on the bottom here on the left, you see a bee. This is a bee that's actually standing on a mat of these plants on water. So this bee is like walking on water because it's standing on these little, all of those little dots are one of these plants. Come on, here we go. Yes, so here's a close-up of one of these very small angiosperms, right, of Lamiaceae. So this is one plant on the left here. And notice you could see, okay, yeah. So on the top it says, each plant produces one complete flower, a flower that has no petals and no sepals and contains male and female sex organs. Yeah, yeah, so here's what the flowers look like. Just as a recall, here, let's move this over here, I guess. Nowhere else to move this. I'll just close it for now. Just as a recall from last lecture, remember, a complete flower means that all the whorls are present. Remember, flowers have 4 whorls. If all 4 of those whorls are present, we call it a complete flower. If the flower is missing one or more of those whorls, we call it an incomplete flower. So this flower, which is at the top here, has no petals and no sepals, missing 2 whorls. Therefore, we can call it an incomplete flower. However, this flower has, this flower has 2 stamens and 1 style, both male and female sex organs. Therefore, even though this flower is incomplete, it is perfect because it contains both the male and female sex organs. And just as a recall, if you're missing one or the other of the sex organs, you're an imperfect flower. All right. Yeah. So that's just the tip of the iceberg to show you the diversity of angiosperms in terms of like really, really big plant, really, really small plant. But there's just a ton. There's a ton of diversity in the angiosperms, which we'll talk about in probably not this time, but next time. But even though they're all super different, right? Even though that big giant eucalyptus tree is huge and that Lamiaceae we saw is super, super tiny, there's certain things that all angiosperms have. And part of the reason that is because the angiosperms are from a monophyletic group. So all the angiosperms that are in existence today, all are derived from a single common ancestor. And now we're going to talk, this is that evolutionary lines in angiosperms. Now we're going to talk a little bit about the different parts of the angiosperm division that we further separate. So within Magnoliophyta, there's different classes. And some classes, the ones shown in green here, have very few members. Very, very few members. The other 2 classes here shown in black have the majority of the species in them. And these are, the long words for them are Eudicotyledone and Monocotyledone. But because that's such a mouthful, even botanists say Eudicots and Monocots, because it's just easier. Eudicotyledone, Monocotyledone. That's even for botanists, we're just like, nope, Eudicots and Monocots. We can handle that. That's easier to talk about. So the Eudicots and the Monocots have the majority of the species in them. Here's examples of some Eudicots. So the Eudicots have about 200,000 species. So that's a lot out of a group that has, what, 366,000? So more than half are Eudicots. Very, very diverse group. These are some examples, but they tend to be trees, shrubs, herbs, you name it. Even cacti are types of Eudicots. And then you have the Monocots, and the Monocots have only about 90,000 species. But some of the species are really important to us because we eat their products. So, for example, a lot of the grasses, remember rice, wheat, barley, corn, those are all types of grasses. Those are all Monocots. But also some other examples are lilies and orchids, palm trees, bananas, etc. So Eudicots versus Monocots. So this is why we're talking about this. This has to go back to the beginning of the lecture where I said you're going to sometimes see the word Anthophyta. So Magnoliophyta, the Angiosperms, right, used to be called Anthophyta when we had it grouped into two classes. We had the Dicots and then, the Dicots were basically everything except the Monocots. So we used to lump it like this. We used to say, okay, well, you have Monocots, right? And then you have everything else, and we're just going to call those Dicots. And that's how we used to have it, and that's when we used to call the flowering plants Anthophyta. But then time passed, people studied more things, and then we realized that that whole grouping of Dicots, so like by grouping all these things in red together, we realized that's really not that right. It's actually more complicated than that. And so then the phylum, the flowering plants or the Angiosperms, we redefined it. We redefined it in that, okay, you have the Monocots, and then you have the true Dicots, which we call the Eudicots, and then the rest, which is all of the things here. So nowadays, this is the way we look at it: Eudicots, Monocots, and then all these things in green, which we just lump together as, say, the basal angiosperms. Basal angiosperms, the rest. Basal angiosperms don't really fit neatly into either Monocots or Eudicots, so we just kind of put them, okay, well, they're not a Monocot and they're not a true Dicot, so they're something else. We'll just they're basal. Put them over there together. So even though that's the case, it's still very common for even plant people, botanists, to use the word dicot instead of saying Eudicot. But just so you know, like even the lab says dicots, it doesn't say Eudicot. But just so you know, if you hear somebody saying the word dicot, they're typically, what they mean is the Eudicots. Most of the time, in this class, for example, we don't talk about any of these green plants. So we say monocot and dicot, even though we mean Eudicot. Okay, let's see here. Yeah, I got time. We're not going to get through it all today, but I got some time. Plus, it's recording, so it'll be on YouTube for you guys. So how are the Eudicots and the Monocots different? There's several ways to tell them apart. Here's some of the ways. So Eudicots have flower parts in 4s and 5s. So remember, all flowers have whorls. All flowers have whorls. But the Eudicots or Dicots have parts in 4s and 5s. So what does that mean? Well, what's an example of one whorl, right? Like petals are a whorl. Notice this plant on the bottom has 4 petals, right? Because it's a Eudicot, they have flower parts in 4s and 5s. This plant up here has 5 petals, right? So these are examples of Eudicots. They have either things in 4, so it would have like 4 stamens or 8 stamens or 12 stamens, or 5s, right? It would have 5 stamens or 10 stamens and 15 stamens, right? Whereas monocots have flower parts or whorls in 3s. So remember, this is our example of a lily, but let's look at the stamens, right? 6 stamens, and that's 3 times 2, right? So 3s. So dicots or eudicots have flower parts in 4s and 5s. Oops. And the monocots have flower parts in 3s. Another way that you can tell them apart is that the dicots or eudicots have pollen grains with more than one opening, right? Usually 3. Whereas the monocots have pollen grains with one opening. Just want to see. Boy, this picture is long, but not, yeah, okay. It's not that bad. We're almost there. Just so you know, pollen grains are really diverse. They can look really, really different depending on what species you're talking about. So this is just a hodgepodge of different pollen grains, what they look like. Here's another way to look at the pollen grains. So this is another hodgepodge. And look at all this diversity in shape, right? Like look at this guy down here. It's like a lizard king or something. But hey, whoa, with this guy here, no, this guy is not from an angiosperm, right? This is not from an angiosperm. Who is that? That's not an angiosperm. That's a conifer. That's Mickey Mouse, right? So that guy, no, we got to get that guy out of there. All right, the rest though are angiosperms. These are all examples of that. But just look at the diversity. Yeah, Mickey Mouse in space. Very, very cool to look at pollen grains. Speaking of pollen grains, so I think in the angiosperm documentary, one of the things that you might have heard of there is that they pollen fossils. So pollen actually fossilizes very, very well. And when you look at pollen fossils, it can actually tell you a lot about the past, in particular, like animal and plant interactions. So one way is pollen grains in fossilized dung, believe it or not, which are called coprolites. If you're interested, you could watch this little, if you click on the link later, you could watch this little thing about coprolites. But yeah, so animal dung, if you have a fossilized dinosaur poop, right, and in that fossilized dinosaur poop is pollen, for example, you'd be like, whoa, these plants have been around since the time of dinosaurs and things like that. Usually it's not dinosaurs, but you get the idea, the fossilized pollen. Can also tell us a lot about the history of our world. So if you think of North Dakota right now, you don't think of it as being a tropical place with palm trees and things like that. But if you look at the pollen fossil record, actually can see that palm trees used to exist in North Dakota. And furthermore, there's even evidence that palm trees used to exist in Antarctica. So this would have been when the continents were together, more towards the equator, right, during the whole different supercontinent type of times. But it's cool that you can like learn these things about the past by looking at fossilized pollen. Okay, so we can tell monocots and dicots apart by the number of parts of their flowers, how many openings the pollen grains have, and then the main difference, hence monocot and dicot or eudicot, is the number of cotyledons. So as you might have guessed, so eudicot or dicots have 2 cotyledons, dicot, 2 dicotyledons, hence why we call them that. So dicots have 2 cotyledons. Cotyledons are embryonic leaves. If you had a lima bean and you cut that lima bean in half, or actually you don't even have to cut the lima bean in half. The lima bean just falls apart in half, right? And so, or a kidney bean or any bean like that. Each of those halves is a cotyledon. So dicot has 2 cotyledons, whereas monocot has 1 cotyledon. Yeah, and just FYI, if you ever see it, for whatever reason in grasses, the cotyledon is called a scutellum because it looks like a shield. So like whatever grasses there, don't worry about scutellum, but you might see that term, like, so here it is here. You might see that term, I think, in lab or something like that. Point is, telling monocots and dicots apart, one way is the number of flower parts, right? Dicots have 4s and 5s, monocots have 3s. One way is pollen grain openings. Dicots have more than 1 opening, monocots have 1 opening. And then another way is these cotyledons. Dicots have 2 cotyledons, embryonic leaves. Monocots have 1 cotyledon. So there's other ways to tell them apart. Another way to tell them apart is how their veins look like. So dicots usually have this kind of net-like vein pattern, whereas monocots have a parallel vein pattern. So can you guys tell the difference? So like, for example, like look at this maple leaf down here. The main way you can think about it is that they have this like central mid-rib that other veins kind of branch off of, right? So it's kind of like a net, like this, right? Like a central thing with things branching off of it for the dicots. Monocots tend to have this, or usually have this parallel venation, right? So all the veins are running parallel to one another. That's another way to tell them apart. These next couple, so we're almost done. Hang in there. These next few slides are going to be really helpful for you guys in lab. So just talking about the venation patterns of eudicots, which is like a net-like or reticulate vein pattern, where monocots have this parallel vein pattern. If you were to cut the leaves in half and look at them under the microscope slide, you can see this. You can see this and be able to tell whether you're looking at a cross-section of a eudicot or a monocot. So, for example, the leaf on the left is from a dicot or eudicot leaf. And it says veins can appear in cross-section or from the side because of the veins net-like pattern. Whereas the leaf on the right side are from a monocot plant, and all of the veins are seen because they run parallel to each other. So here, let me see. This could help here. So the idea is, like, if you have veins in your leaf, so like let's say this is veins, and let's say these veins are in a net-like pattern, right? Like this, right? If I were to do a cross section through this and look at it like under the microscope slide, you'd see like maybe this maroon color would be like coming at you, right? But then these would be kind of going off to the side, and these would be kind of like going off to the side. And that's what we see on here. So let me put this, right, so on the left, remember that's a eudicot. Here is a vein. The circle is showing a vein coming like right at you, right? Like, so coming like straight at you like this. Whereas the rectangle is showing you one of the veins that kind of is branching off to the side because of its net-like pattern, right? So like here's one vein coming right at you, and then one's branching off to the side. You're only going to see like part of it if you were to do a cross section. However, with the monocots, all of the veins are parallel. All of the veins are parallel, or like this, right? Parallel to one another. So if you were to do a cross section through them, you would see the tips all coming at you because they run parallel. And that's what this is showing in the circles, right? Each of these looks kind of like a little face, is a vein. They run parallel, so you can see the tips of each of them because of that. As this will be helpful in lab. Similarly, monocots and dicots, the primary vascular tissue looks different. So if you were to look in this stem, the eudicots have the vascular bundles arranged in a ring. And this is stuff you'll cover in lab. vascular bundles arranged in a ring. Monocots have vascular bundles scattered in the stem. So I'm not going to spend time on this because you guys will see that in lab, but this is a good resource for you guys to have in lab. And if you're looking at the primary tissue in the root, the Eudicots have their vascular tissue in the root centralized in one cylinder in the very, very middle, whereas the monocots in the root have this vascular cylinder that looks like a ring. Again, I'm not going to spend a lot of time on that because you will see that in lab. Okay, we got like one more slide. The other thing is that, so this whole end of this lecture is talking about telling Eudicots and monocots apart. Eudicots can have true secondary growth, right? So they have wood for those species that do get secondary growth. However, monocots rarely do. So you can see here, this is an example of a Eudicot on the left. Here is bamboo, or sorry, this might be sugarcane. Well, either way, it's a grass. And yeah, okay, it gets hard and rigid, but it's not due to like massive secondary growth, right? You're never going to see a bamboo that's like 4 feet wide, right? So monocots tend not to have a lot of secondary growth, meaning secondary xylem rather. Okay, so that's covering that. As I said before, at the end of this lecture, and again, these lecture slides are posted on Blackboard as well. You have these nice summary tables. So here is a summary of how the Eudicots and monocots are different, the things we just kind of went over. Here's a nice summary table of the cells that we talked about at the beginning of the lecture, of the xylem and the phloem. And then this is just for, I don't know if you will need to use this slide ever. This goes a little, but the point is at least it's here. So here's another summary table of tissues and cells to help you get organized. This might be useful to use in lab. Okay. Yeah. Oh, this is good though, because it tells you the difference between those xylem and phloem cell types that we went over. So I like having summary tables, kind of puts it all together. Okay. Thank you guys. Next time we will keep going with the angiosperms. We only have 2 lectures left, so sorry for barreling through it today, but I finally solved my technical difficulties. We won't be having those anymore. And thank you guys so much. And yes, see you guys. Thank you so much. If you have any questions, email me, or I can answer any questions now if you want as well. When is the lab exam? You'll have to check the syllabus. I think it's supposed to be the last day of lab. So you should check the syllabus for that, or ask Rajan. Yeah, May 1st sounds correct. Sounds correct, because that would be, I think, the last day of lab. Yep. Oh yeah. So the lecture exam is, you can look at the exam schedule on the records page, but it is, I believe, scheduled for the, oops, I'm in April. I think it's scheduled for the 5th or the 12th. It must be the 12th, because I think that's when finals week is happening. The final exam, remember, it's not cumulative. It's just going to cover the angiosperms. It's not going to be, out of the 3 exams, it's definitely the easiest of the 3 exams. There was a question about phenology assignment. I said in the beginning of the class, still kind of figuring that out. The original due date was for the last day of class, but I'm going to push that due date back, and I'm still figuring out what that's even going to look like. Any other questions? Yes, thank you. And I will post the the link to my dissertation, so you guys can watch if you want to, happening this Monday. All right, guys. Thanks, and I will see you next week. Have a great day, guys. Bye.